Hello, everyone. So a few people are already joined the webinar room, but we will wait 5 more minutes so everyone has the chance to join this webinar, and we will start in 5 minutes and 4. All right, then see you in 5 minutes. Oh, I think we lost Elisa, but hopefully she will join again in a second, and then we will start the webinar. Hello.
Good morning. Hello, everyone.
Will Elisa join your office?
Elisa is in her office. She will join soon, I think.
We lost her. Oh, like you. Hi, you were a few seconds away. I'll add Elisa again, so she can join the presentation.
I'm not familiar with this setting, so
No worries. We tried this program yesterday. Sorry for the circumstances, but Elisa will join. I just sent her the co-presenter link again, and so we can start very, very soon. Here you are again. Hi.
Yeah.
No worries. So I think we can start now. A few more people have joined already, and yes, hello and welcome to our fourth webinar of the Permeability series in 2022 with the topic Human Lactobacillus Biosurfactants as Natural Excipients for Nasal Delivery of Hydrocortisone. My name is Yannick, and I will be your host for this webinar. On today's agenda, we will take a closer look on the publication from Corazza et al. We are happy to have our second external speaker with Elisa Corazza here at the Permeability series. She's a PhD candidate in the group of Professor Barbara Lupi, you have seen shortly before, from the University of Bologna in Italy. Her aim of the PhD project is to develop innovative strategies to improve local and or systemic treatments by increasing drug penetration and permeation through the nasal mucosa. Moreover, part of the research considers are the impact of probiotics and their metabolites on drug absorption and penetration. In her presentation, she will focus on the experimental setup with the preparation of the barrier and how you can use it for the nasal delivery. If you have detailed questions regarding the analysis, Elisa is happy to answer it in the Q&A session. Before we start with the webinar you've been all waiting for, a few things regarding the organization. We have opened a Q&A window on the right side, the chat. You can write for the whole community or a private message. And we will answer them at the end of the presentation, and we'll also provide the slides and a link to the recorded webinar. Without further ado, I give the word to Elisa. Enjoy the presentation and see you at the Q&A session again.
Thank you, Yannick, for the kind introduction. I'll share my screen so that everybody can see the presentation. Okay, I just ask you if you can see my screen and, of course, the presentation. Can you see it?
I can see it, yes.
Very good. So before starting, let me thank Fabio for having invited me as a speaker to this Permeability series. And then I think we can start. So today I'm glad to introduce you the results of our work into the use of Lactobacillus biosurfactants as innovative and natural excipients for nasal drug delivery. During my speech, I'll do a very brief introduction just to clarify the aim of my study. Then I will go through the materials and methods employed to obtain the results, and I'll finish summing up the main conclusions. The nasal route exhibits a lot of advantageous features, including ease of administration, avoidance of both needles and first-pass metabolism, as well as a high vascularization with a consequent increase in drug uptake and absorption. Moreover, it offers a direct access to the cerebrospinal fluid. However, it also presents some shortcomings, such as the restricted surface of the nostrils, which strongly limits the applicable volume of formulation and affecting mainly those drugs characterized by a low solubility in aqueous medium. Then drug absorption and also effectiveness are also hindered by the architecture and composition of the nasal mucosa because it is made of two barrier systems. The first is represented by the airway epithelium and the second by the mucous layer that lies on it, with the latter being subjected to a continuous flow and turnover due to mucociliary clearance. So these considered together with the growing interest toward nasal drug delivery, there is the need to find out ways to overcome the limitation posed by such an administration strategy. The inclusion of chemical permeation enhancers like surfactants in a dosage form is considered a very straightforward approach that is frequently used to improve drug absorption across biological barriers, including the nasal mucosa. And among these molecules, biosurfactants are gaining interest because of their being natural products, and so they better suit the current trend of the scientific community to look for more eco-friendly materials. And in fact, biosurfactants are microbial molecules produced as secondary metabolites, and they exert different physiological functions inside the bacterial cells, but they are of pharmaceutical relevance because of some of their features. In fact, apart from being green products, they are characterized by a low toxicity and high biodegradability. They exert surface and emulsifying properties, and they were found effective at extreme temperatures and pH values. Recently, two biosurfactants isolated from different human lactobacillus strain, BC1 and BC9, were found to be useful as a permeation enhancer in transdermal drug delivery, and because they both increase drug solubility and permeability across the skin. And as a result, the purpose of this work was to evaluate the biosurfactant produced from the Lactobacillus strain Gasseri BC9 as a potential green excipient to improve hydrocortisone absorption across the nasal mucosa. And precisely, BC9BS properties were compared to those of other frequently used surfactants, such as cocamidopropyl betaine, CAPB, and the alpha-tocopherol polyethylene glycol, TPGS. First of all, in collaboration with the microbiology lab of Professor Vitali at University of Bologna, we exploited a very straightforward approach to extract and isolate the lactobacillus produced. And in particular, we focus on the fraction of biosurfactant that is bound to the cell surface. And just briefly, 100 milliliters of an overnight culture of lactobacillus were inoculated into 900 milliliters of MRS broth and allowed to grow for 24 hours. Then cells were harvested by centrifugation. Cell pellets were washed twice in sterile water and resuspended in PBS for 2 hours so that to allow the release of the biosurfactant that is bound to the cell surface. The suspension was then centrifuged and purified and filtered. And the cell-free supernatant was purified through dialysis, and the purified biosurfactant was reduced into powder by freeze drying. Thanks to previous analysis, it is known that the biosurfactant is composed of a mixture of peptide-like molecules containing hydrocarbon chains of different lengths, and in their peptidic portion can be found amino acid residues like histidine, valine, and threonine. Hydrocortisone solubility was studied by dissolving an excess amount of hydrocortisone in buffer pH 5.5, which resembles the pH of the nasal cavity, in absence and presence of the three surfactants used at concentrations below or above their critical micelle concentration, CMC. After 48 hours at room temperature, the fraction of undissolved drug was removed by centrifugation and subsequent filtration, and the drug concentration in the supernatant was determined through HPLC assay. Data show that below the CMC, all tested surfactants were able to improve hydrocortisone solubility with respect to the control. But such an increment was even more noticeable when surfactants were used at a concentration above their CMC. And this is reasonably due to the drug entrainment into micelles. But overall, we can state that BC9-BS was able to improve hydrocortisone solubility and that it could interact with the drug both as a single molecule and as micelle. Along with its solubility properties, BC9-BS ability to interact with the main component of the nasal mucosa was also investigated. In fact, surfactant's ability to interact with mucin was studied through turbidimetric measurement of a suspension containing a dialyzed and lyophilized mucin in presence of surfactants at concentration below or above the respective CMC. The absorbance ABS of the mucin dispersion itself and the surfactant suspension were measured as references because here data are reported as the percentage ABS increase in the sample in presence of mucin with respect to the same sample without mucin. And what comes clear from the graph is that below the CMC, only BC9-BS and CAPP were able to interact with mucin, whereas no percentage ABS increase was observed for TPGS, and this is reasonably due to its non-ionic nature. But interestingly, when surfactants were used at a concentration above their CMC, none of them was able to interact with mucin. And so we hypothesized that this was due to the micellar organization, which probably hides those reactive groups that were previously available for mucin interaction. Lastly, surfactants' ability to improve hydrocortisone absorption through the nasal mucosa was investigated through in vitro diffusion studies across different in vitro barriers. Of course, the first was the PermePad barrier, which being composed of phospholipid sandwich between two low layers, it is known as a biomimetic membrane. And this model was chosen because of its useful features. So apart from its cost effectiveness, because it is resistant to a wide range of pH, and because it is compatible with different co-solvents, surfactants, and simulated fluids. Moreover, it is able to mimic not just the cell membrane, but also the composition, the tissue morphology, we can say. The second barrier was based on the same PermePad model, but enriched with a reconstituted layer of mucin, a mucin that was previously dialyzed and lyophilized, and the suspension was dripped, so released on the top of the PermePad and left to equilibrate a few minutes prior to sample addition. This way, we developed an in vitro model capable of taking into account not just the airway epithelium, but also the mucous layer that covers the nasal cavity. And the last model was represented by the sheep nasal mucosa, precisely the tissue excised from the nasal turbinates. Here I reported the apparent permeability coefficients of hydrocortisone, so our model drug, across the different in vitro barriers, in absence and presence of the three surfactants used at concentrations below or above their CMC. Firstly, when the permapad barrier was used, so figure A, only TPGS and BC9BS were able to increase hydrocortisone permeability with respect to the control at both concentration tested. Differently, when the permapad and mucin model was used, so that are depicted here in figure B, all tested surfactants were able to significantly improve hydrocortisone permeability with respect to the control. And I would like to highlight that there wasn't a significant difference between each surfactant when used at concentration below or above its CMC. Finally, to test whether our in vitro models were able to predict and foresee BC9-BS activity in vivo, in vitro diffusion studies were performed across the sheep nasal mucosa, but only in absence and presence of the biosurfactant at a concentration below its CMC. And as expected, the use of the biosurfactant allowed for a great improvement in hydrocortisone permeability with respect to the control. To conclude, we can state that we obtained promising results because our biosurfactant was able to increase both hydrocortisone solubility and permeability across the different in vitro barriers that we used in this study. It is worth saying that the biosurfactant was found effective even when it was used at the lowest concentration tested, which was exactly the same at which the biosurfactant was seen able to interact with the main component of the nasal mucosa. So overall, it seems that BC9-BS could be a promising alternative to chemical surfactants in the nasal drug delivery field. Before moving to the QA section, let me thank my supervisor, Professor Barbara Luppi, and all the people who contribute to this work. So all my colleagues from the drug delivery and microbiology labs at University of Bologna, as well as Professor Massimiliano Pio Di Cagno from the University of Oslo. I would like to thank again Fabio for giving me the opportunity to be here today. And of course, I would like to thank all of you for the kind attention.
Thank you very much, Elisa. Very interesting presentation. I just changed the layout again, and we just got the first questions during the presentation. It was, the question is, why do you choose Lactobacillus in first place? Why not others?
Yeah. That's a good question, good point. So thank you. It is known that lactobacillus have a nick into the nasal cavity, so it is reasonably starting from this strain, even though it's not the most representative of the nasal cavity microbiota. And then the other reason was that in our lab, there is we were already working on this strain, not just BC9, but also others. So we already had information about the physiochemical features of the biosurfactants and also other metabolites.
Okay, that's great. I have a question regarding the analysis. What was your approach to analyze your data? Because there are several approaches: area under the curve or the recipe app. What have you done there?
Yeah, we analyze all the samples from the permeation study through HPLC, measuring the area under the curve and then plotting it. And we represent it as an apparent permeability coefficient.
Okay. Yeah, very good parameter.
So we studied, of course, both the flux, the apparent permeability, but then we prefer to show the apparent permeability coefficient that is usually the one that is shown and that takes into account also small differences in drug concentration.
Okay. Is there any other question? Yes. Do you plan a KCO2 assay to determine if there's a paracellular transport?
No, we just studied the passive diffusion because also the sheep nasal mucosa, I presented it as an in vitro model because we weren't able to maintain the tissue alive. So also the sheep nasal mucosa allowed us to just study passive diffusion.
Maybe a little add-on. Pia, maybe you have to visit our next webinar in November because there we will present a paper where it goes, or where the topic was the paracellular transport with PermaPad. So maybe you can determine the paracellular uptake with PermaPad as well and mucin. But then you have to join the next webinar. And we have another question from Cosmin. Have you tested also other drugs? And the second question was, do you observe the same increase of permeability?
We are planning to perform similar experiments to these with also other drugs, and we use the same model, so PermaPad and mucin model with, for example, carbamazepine, so another low water soluble drug, and we observed the same. I mean, the model worked the same way, so we observed a decrease in permeability in presence of mucin with respect to the same model in absence, but it was slightly different because we used not biosurfactants or other natural surfactants, but chemical, so synthetic surfactants, but the result was the same.
Great. Then we have another question. This is for us, for Inume. Do you plan to add mucus on PermaPad in commercial kit? This is what we're considering right now, but it's not commercially available. But what we are working with a company together, maybe we will, and they will have, or they have, synthetic mucus you can add by yourself. So this is not a complete kit, but you can do it by your own. But we work on a commercial kit, so you already have the PermaPad coated with mucus. But there is a possibility to do it by yourself. And if you're interested, you can contact me afterwards, or I will write you an email and give you the contact details for the mucus company. All right. I will write you an email. Great. So as I said before, we are planning a new webinar and the last one for this year in November. Stay tuned and follow us on LinkedIn, so you will get all the information about the date and the topic and the speaker. We always post there. And I already shared the slides. You can download it, and you will receive a link from Webinar Ninja with the recordings of this presentation. And if you have further questions, you can write every time us or Elisa directly, and then we are happy to help you. And we are looking forward to meet you at our next webinar from the Permeability series.
Thank you very much, Nick.
Yeah, you're welcome. Great presentation. So everyone have a great day, and we will talk soon. Bye-bye.
Bye.